Today we will discuss about enzyme inhibition and basic mechanism of this enzyme inhibition. The main or most important enzyme inhibition are competitive, non-competitive and uncompetitive inhibition. In detail of these three that we will understand with the help of this equation and description. So basically what is competitive inhibition? In competitive inhibition, substrate is generally attaching over the enzyme, but here inhibitor molecule that is structurally similar to the substrate, and so this inhibitor is competing for the active site over the enzyme. So basically inhibitor being structurally similar to the substrate is compact over the substrate, and so it is making a competitive inhibition. So normally enzyme plus substrate making ES complex, and from that product will be generated, but in this inhibition inhibitor is attached over the enzyme, so making enzyme inhibitor complex, and from that product will not be generated. So inhibitor binds to the free enzyme only and compacts with the substrate. So increasing substrate concentration overcomes the inhibition by inhibitor. Simple. If there is a equal number of substrate and inhibitor, then equal chances of that inhibitor to attach over the enzyme. But if you are increasing substrate concentration more, about 75 or 90 percentage, then more substrate will attach over the enzyme and inhibitor attachment will be less. So if you are increasing substrate concentration, then definitely this inhibition will be decreased. Then next, that is non-competitive inhibition. What happen in this non-competitive? Substrate is attaching over the different site over the enzyme and inhibitor is attaching over the different site over the enzyme. So there is a no similar site, so there is a no correlation with the structure. So first, in this competitive, you have to remember inhibitor is structurally similar to the substrate, but in non-competitive inhibitor is not structurally similar to the substrate, so it is attaching over the different site. So when it is attaching over the different site, then it is inhibit this enzyme to convert this substrate into the product. So in detail what happen, enzyme plus substrate making ES complex, and from that product is generated, but here inhibitor is attached either over the enzyme alone, or inhibitor is also able to attach over the ES complex. So ultimately enzyme inhibitor substrate complex will be generated, but product will not be generated. So inhibitor binds to the free enzyme or ES complex. Increasing substrate cannot overcome inhibition. So that is non-competitive. Now next, that is uncompetitive. It is most interesting is that inhibitor is not able to attach over the enzyme only, but inhibitor is able to attach over the ES complex only. So that is the difference between this uncompetitive and non-competitive. Uncompetitive inhibitor is not attached over the enzyme molecule only, it will attach over the ES complex. So it is attaching over the ES complex and making enzyme inhibitor substrate complex. So inhibitor binds to the ES complex only. Increasing substrate concentration, it will favors the inhibition by the inhibitor, means more substrate is added, more ES complex will be generated, and more inhibitor is able to attach over the ES complex. So that is about the basic mechanism of this enzyme inhibition in relation to competitive, non-competitive and uncompetitive inhibition. Now further we will understand from these different plots or direct graph and double reciprocal graph. So what happened in this competitive inhibition? So normally this type of reaction, that is we can understand from this direct plot, substrate concentration, velocity, means velocity that is over the y-axis. From that we can get Vmax, maximum velocity, and Km value. Km value, that is Michaelis constant, and that is called as substrate concentration at half maximal velocity. Now it is interesting to understand about this different inhibition and what happened to this Vmax and Km value. So it is frequently asked in the Viva and MCQ also. So in competitive inhibition what happened, Vmax will remain same but Km value will increase. So when Km value is increasing, that is inversely proportionate to the affinity, means Km value is increased, then enzyme has less affinity to the substrate. So that is Vmax will remain unchanged in this competitive inhibition, but Km is increased, means decrease the affinity for the substrate when inhibitor is attached over the enzyme. Same thing we are getting in this double reciprocal plot. It is very easy to get this double reciprocal plot when that limited concentration of substrate is present. So this double reciprocal plot is giving the similar type of value, that particular both the line intersect at the y-axis where we can get the Vmax. So Vmax will remain unchanged, but Km value is increased when competitive inhibitor is present. Now what happened in non-competitive inhibition? Vmax we are getting and Km value from this direct plot, but in the non-competitive inhibitor is present, velocity will decrease, that is maximum velocity is decreased, while Km, that is Vmax is decreased, while Km is remain unchanged. So that both line is intersect at x-axis where we are getting the Km value. So Km, that is Michaelis constant, that is remain unchanged in non-competitive. So you can remember that no change in Km value in non-competitive inhibition. Now next we have seen about the uncompetitive inhibition. In uncompetitive inhibition, interesting is that Vmax is decreasing but Km value that is decreasing. So Vmax is decreasing, that is normal thing in this uncompetitive, but interestingly that Km value is also decreasing, means that inhibition, uncompetitive inhibition is present, then affinity of enzyme towards the substrate is more increasing. So ultimately both the line is not intersecting to each other at any y or x axis, but both Vmax and Km value is decreasing in uncompetitive inhibition. So that's all about the three most important enzyme inhibition: competitive, non-competitive and uncompetitive. Further, their clinical significance we will look in the next video. So that's all about competitive, non-competitive, uncompetitive. You have to remember Vmax is unchanged in competitive, Km is changed in competitive, non-competitive, Km that is remain unchanged and Vmax is decreased, but both are decreasing, means Vmax and Km both are decreasing in uncompetitive inhibition.